So in the US, there's essentially three major types of peanuts. You have the runner type, the Spanish type, and the Virginia type. So in Georgia, the main peanut that we produce is a runner type. And runner peanuts tend to be a little bit smaller. The peanut is a little bit smaller. They have a good yield, so they produce quite a bit. If you go further north up into Virginia, you get the Virginia type peanuts, which are the larger, more confectionery type peanuts. And if you go further west out to New Mexico, you get the Spanish type peanuts.
Georgia is the largest peanut growing state. Its farm gate value is a little over $825 million a year.
So it's a major part of the agricultural portfolio in the state of Georgia. Thus, it's a major part of the research portfolio at the University of Georgia.
The peanut is a great plant-based source of protein. Peanuts contain somewhere around 24 to 26% protein. They contain about 50% lipids. And in fact, we also have a class of peanut called high oleic peanuts, which are bred specifically to maintain a high oleic acid content of around 80%.
Studying the genetics of crops and mapping their genomes is important to agriculture because in particular, we have a growing global population, and in order to feed that population, we have to improve the productivity of the plants that we grow. There's only so much arable land in the world, and we've already essentially used all of that arable land, and so we really have to improve crop productivity, of course, while maintaining the nutritional content and also, of course, satisfying consumer preferences. Once we understand all of the genes in a genome and how they contribute to the development and the form and the function of a plant, the more efficiently we can go about actually modifying those genetics to improve the plant productivity as well as nutrition and taste, perhaps.
Understanding the genes that control yield, the genes that control how a plant responds to environment, understanding the genes that control how a plant responds to different stresses like a fungus or a virus or insects or heat or drought or too much water will allow us to engineer plants that are better adapted to a broader range of growing environments, will yield better, and importantly also be more nutritious for the people who consume these plants.
We've been working on genetic resistance for aflatoxin contamination for many years now. We have made progress, though, in making selections for lines that do have reduced levels of aflatoxin contamination.
We're doing some work where we're combining hydrogen peroxide treatment with UV radiation to see if we can reduce the aflatoxin contamination. People have done a lot of other chemical treatments as well as some physical treatments to reduce aflatoxin.
Now we have a better understanding of the genes that make up peanut, and it allows us in the future to be able to engineer the peanut to be more productive, to be more resilient against climate changes or variability, to be resistant to various pathogens that attack it, and to engineer it for quality.